Greetings everyone, my name is Fizree, and today I will share with you my research on optimization of protoplast, isolation for alpine, vitro-derived and mesocarp. So a bit of the background on the protoplast. Protoplast is a naked plant cells with the cell wall removed. So this will make it easier to manipulate DNA inside the cell. Protoplasts are the closest that we have on the cell-based assay. It is much more beneficial for in vivo gene function study and also gene editing for concept study. So on the methodology, how we do it. So the sample first need to be prepared. The mesocarp need to be sterilized first before proceed with the experiment. For the in vitro, for the leaf, says it was derived from in vitro leaf. So sterilization doesn't need to be done, doesn't need to be carried out. The sample need to be sliced thinly to increase the surface contact area with the enzyme mixture. And since the leaf have wax cuticle, it need to be removed using scalpel before proceed with the experiment. After the sampling was carried out, plasmology was done in the mannitol solution. But then the samples was incubated in enzyme mixture and put under vacuum for 30 minutes before incubation for 14 hours to improve the penetration of enzyme mixture into the tissues. Then we proceed with washing and purification to recover the protoplast. So we did optimization using a mixture of enzyme involving the cellulase R10, the enzyme R10, the drisalase, and also petolase Y23. At the end of the study, we found that a mixture of 3% of cellulase R10, 1% of enzyme R10, 1% of drisalase, and 0.1% petolase Y23 has provided us with the best yield for the in vitro derived for protoplast from in vitro derived leaf with up to 2.5 times 10 power of 6 cells per gram fresh weight after 14 hours of incubation. So then we use the same enzyme mixture for the isolation of protoplast from alpha mesocarp, and we found 2 hours of incubation is enough to yield around 3.98 times 10 power of 6 cells per gram fresh weight of protoplast from mesocarp with up to 85% of viability. So here we can see as the image of protoplast stained with CFDA. The green fluorescent showed that this is the viable protoplast. So in conclusion, the mixture of cellulase R10, mesosome R10, drisalase, and petolase Y23 enable efficient protoplast isolation from a mature alpine tissue. So this ideal enzyme incubation period will require 14 hours of incubation for efficient isolation of protoplast from in vitro derived leaf of alpine, while it only need 2 hours to isolate the protoplast from alpine mesocarp. So for the recommendation, healthy condition of source tissues are important and significant in determining the end yield of the isolated protoplast. Also, the type of enzyme included in digestion mixture and their concentration affect the yield and viability of isolated protoplast. Thus need to be determined empirically for every different tissues used. That's all. Thank you.